This conference will now be recorded.
Yeah, to make it safe, I won't, maybe at the very beginning, I will switch on my camera, but then I will make the presentation without, just to be to on the safe side with the internet connection.
Okay, that sounds good.
Yeah.
Sounds perfect. We'll wait maybe a minute or two. I think most of everyone is joining right now, so maybe just 1 minute or two, and all right. So I think in the interest of time, we should get started. I know a few people might be out on vacation, and others might be joining a little later. So I see a few of you are joining in. Thank you everyone for calling in. Today we have our invited speaker, Dr. David Rouquie from Bayer Crop Science. David is an active E-Star member, and he's been with Bayer for over 10 years in different roles, from study director and most recently to early safety representative. Dr. Rouquie has led research and authored several publications in the in silico and molecular predictive toxicology space. To this end, today he will be presenting his talk titled Interplay Between Chemical and In Vitro Biological Spaces, Opportunities for Toxicity Prediction and Compound Denovo Design. So with that, I will turn it over to you, David. Thank you so much.
Thank you to you, Carolina. And I just want to say hello to everyone, and switching on my camera, but then I switched off for to save my internet connection. Let's put it like this. Thanks a lot for the invite of giving me the opportunity to give this presentation. And today I would like to guide you through some work we have been doing over the last few years, and I will somehow start. I would call it the American way. I would start by making the acknowledgement first, and I would like to thank the team and acknowledge all the people who are involved in all the things that I'm going to present you today. Starting with the colleagues who are working in my team. So from the left, Oscar was postdoc, and Ingrid was doing a PhD with me, and then Benoit, who did some internship student. He was internship student in the lab. And then there are some other colleagues from Bayer Crop Science and Bayer Pharma, and just to cite two of them, so Oko Clevert and Jörg Wichard, or actually what I would call the hardcore machine learner in the team, and they did support a lot of the work that I'm gonna present you today. So in terms of agenda, I will provide you with also a view of exploring biological spaces, and I'm gonna come to this definition about what I mean by biological spaces later on. And then I will continue by providing some example of what we have been doing in terms of Denovo design of EAT molecules using biological space data. And then I will end my talk by a proposal for a cross-sector collaboration. So, as you know, for us in agrochemical industry, we have to design molecules which are sorry, I just need to minimize the screen. We have the aim of developing and finding molecules which have very specific types of activities either toward the target organisms, could be fungi, pests, or weeds. And at the very same time, we need to make sure that this activity, which is still there, won't be too high in terms of off-target organism. Could be, of course, human, mammalian species, or invertebrates, or everything dealing with ecotoxicology. So my talk will be very much focusing on human safety, so the toxicity toward humans, and to do so, actually, we go for several steps of chemical and the key thing is it's a multi-domain chemical optimization when it comes to safety profile optimization, when it comes to agrochemicals molecules. And typically, when we are getting a hit from an high throughput campaign, we have different type of profiles of the molecules. Typically, we are having a rather good activity toward the biological target. And then we have different parameters in these profiles which have to be optimized. And just to name some of them, so the dynamics properties, the toxicokinetics properties of the molecules coming jointly together with the physicochemical properties and some other parameters have to be optimized in a way to get a registrable molecule that can be launched on the market. And all those steps of multi-dimensional chemical optimization rely and make use of different types of tools when it comes to toxicology, starting from data visualization, of course, data sharing and data access with the other colleagues from the chemical departments or the ecotoxicological department. It enables us to go into data-driven decision-making. We are, of course, also going into mechanistic knowledge information. And at the end, we are applying some data modeling techniques, relying on machine learning or PBPK modeling, just to cite two of them. So before jumping really into the real case, I just want to make some clarification about what we mean and about spaces in this context. So you might have seen this this slide many times, but it's a way one can use to describe the how an adverse outcome is developed into a laboratory animal. So you start with the chemical has to enter the organism, reach a certain tissue dose level, and then hit a target, and then induce some changes which depending if the time and the dose of exposure are high or long enough, might translate to an adverse outcome in the animal. So for us in particular, when it comes to the early evaluation, early characterization of our young molecules, of course, we need to find surrogate type of systems to inform about the profile, the toxicity profile of these early candidates. And there are several options there. And today, during my talk, I will be focusing on two main ways. So the ones dealing with the identification of the molecular targets, or the mobilization of the molecular targets and looking at the molecular pathway or the first cellular changes, in that case, in some in vitro test systems, in order to first take into consideration the throughput, the number of chemical entities which need to be evaluated that cannot be done in laboratory animals, and also the amount of the molecule which is at this stage available, which prevents, even if we would think of doing that, it would prevent us to do that just simply due to the too low amount of molecule which is available at this stage. So thinking about in vitro assays, we've been looking at some effects in vitro in terms of gene expression signatures using different technologies. So we've been doing, and that will be the thing I will present you shortly after, about the work we have been doing on L1000 technology datasets from the Broad Institute. In the lab, we are using the GoScreen technology, which is a DNA-based transcriptomic setup. But there are also some RNA-seq, so TempoSeq in particular. So this is the first, let's say, endpoint that can be measured from in vitro test systems. The other one I will discuss today is about in vitro cell image. And I will be talking mostly about the cell painting technology. And now comes to this definition of space. So when I will be doing my talk talking about chemical space, it will be referring to a set of different chemical entities. And when I will be talking about biological spaces, it will be the results obtained of the effects of the molecules into an in vitro test system. That's what we call the biological spaces. So having said that, when it comes to prediction, since it's the topic there, of course, we need to get the appropriate ingredient to do so. So the in vitro bioactivity information is of course very important, and this in a dose response manner to determine the point of departure. But something which is also extremely important is about having information about the bioavailability or the ADME properties of the molecule to make this translation between the in vitro test systems into the in vivo situation, where you see, even in terms of units, you move from micromolar, let's say, to milligram per kilogram per day to make this prediction. So just to cite two examples of things which have been published so far. In terms of bioavailability prediction, we have this the very first paper for Wetmore et al., where this reverse dosimetry aspects was performed in order to make use of the in vitro data in order to predict the in vivo outcome in the next step. And I would like also to highlight another initiative that has been done by our colleagues in pharma to make prediction of the oral bioavailability, in that case in the rats, which was a combination of machine learning and machine learning based on chemical structures and QSAR, let's say, and PBPK modeling in order to make this prediction possible. So having said that, and having the tools clearly named, so let's now have a look at what type of data could be available to make this tox prediction. There, assuming that in this triangle, the x-axis inform about the amount of information which is available. So from the bottom to the top, you get less and less data available for this. So we start at the bottom from the structural information. So up to now, globally speaking, the industry is dealing with chemicals or libraries in the range of millions of chemical entities, and now with the new developments, it will go certainly much. The numbers will increase dramatically. Then moving up, moving up in terms of data informing about the chemical entities when it comes to the toxicity. So we have a series of on and off target assessments. So in our case, we have, of course, I mean, just again to cite a few of them. So some data related to the genotoxicity or mutagenicity in the case of AIMS, some data about steroidogenesis for the endocrine-related toxicity, some binding assay toward target transactivation assays. When it comes to some key receptors or key enzymatic enzymes which are involved in some tox outcomes. Moving upwards, we have more integrative biological responses that could be then in that case high throughput transcriptomics or cell painting. And then already there you see that the numbers are decreasing. And to reach the tip of this triangle where we have the in vivo outcomes or the in vivo tox data, which are even in our case at Bayer, where we can combine because we have the two division into the same company. So the pharma division and the crop science division are jointly working together. Even if we think about joining forces there between pharma and crop science, still the number of in vivo tox data are still limited, or to say it a little bit differently, I mean, there are plenty of data, but not all of them are available in a decent machine readable format so that we can make use of them. So up to now, what is classically done in terms of prediction, we can start from the chemical structure. There are plenty of QSAR models in particular, or machine learning or rule-based methods which enable us from the chemical structure to predict some properties of the molecules. We have been publishing some works showing that using biological responses, we can infer about some targets of the molecules, and this is going to be exemplified later on during my talk. And there might be an opportunity to move from these more integrative biological responses to predict in vivo outcomes in the near future. There are one paper in particular which has been already published on going into this direction, but yeah, there must be still a lot of work to be done to make this really possible. So now I'm kind of finished my intro part. I will move on to these examples of the values of exploring biological spaces. And again, taking one example on high throughput transcriptomics and another one on cell painting. So to do so, I will start with a work we have been doing using a public data set from the MIT Broad Institute. It was published end of 2017, where the team has disclosed a huge set of transcriptomic data derived from many different molecules. So they were talking about 20,000 different molecules in many different cell lines. And there were some genetically perturbed cell lines as well, so loss of function, gain of function, and and even some biological treatments, biologics in the sense of in the pharmaceutical world. And so in this work, very nice and cool work, the idea was to show, I mean, the value of this type of measurements derived from in vitro test system, in particular with different cell lines, human tumor cell lines. And so what we have done was to make use of those data. So 20,000 in total, 80 cell lines. We ended up with something like 350,000 signatures. The technology was the so-called L1000 technology, which actually provide the information about the level of expression of thousands of genes, landmark genes in the genome. And we went through a step of data processing where we took all those data and we made some filtering, let's say, to make to determine a minimal set of data which was sharing the same type of study design. So for this, we extracted all the information related to all the compounds for which the molecule were treated at 10 micromolar for 24 hours. And we ended up with a kind of full matrix of results for 9,000 compounds, which were almost all of them tested in 8 different cell lines. And this was the basis of the work I'm going to show you as a starting material. So 8,000 molecules in 8 different cell lines. And now come the first slide of the chemical space. So the chemical space, this is the chemical space. So this is a t-SNE plot where every dot on this 2-dimensional plot represents a chemical entity. And something to bear in mind is that in this type of representation, the closer the dots are, the closer the structure of the two molecules or the molecules are together. So if you pick molecules in the same cluster, they will share structural similarity, whereas if they are far away in the plot, they will be much more different. So this is the chemical space of the 9,000 molecules that were used in this exercise. And similarly, we did a representation of the biological space. So in that case, it's the 48,000 signatures of the 9,000 molecules which were used in the different cell lines which are listed there, derived from different tissues. So from breast, the prostate, the lung, the kidney, from the skin, the lung, and the colon. Okay. So each dot in that case is one signature. The closer the signatures are, the more similar they are, or the more similar they are. And on top of that, we have overlaid the colors indicates in which type of cell lines this signature has been generated. Okay, so one of the first thing we did was to have a look specifically at the results obtained for these 9,000 molecules in the cell lines which were derived from the same tissue originally. So we started to have a look at the results of the gene signatures from the 2 cell lines derived from the prostate, VCAP and PC3, and the 2 cell lines which were derived from the lung. And those are the results we did obtain. So in brown, you see the result from the VCAP, and in blue, you see the result from the PC3. So those cells are derived again from the prostate. And something which was kind of extremely surprising to me was to see how diverse and how little the gene signatures were overlapping between the 2 cell lines. So meaning that we were looking at very different biological spaces when it comes to 2 different cell lines derived from the same tissue, the prostate. So this was the first example, and we did observe the very same type of phenomenon when we look specifically in the 2 cell line derived from the lung, where I mean we could see some overlap, but very little compared to the overall signatures from the in the entire in this data set. So that was the very first exercise we did. Afterwards, we played a little bit with the metadata information about those molecules, and to make me, I tried to make it very understandable for you. So we collected some information among the 9,000 molecules about the molecules which were known to have been tested toward specific targets. And yeah, if you can please put you on mute, because I just have the, sorry for that, but I can hear people speaking. So in that case, we collected the information among the 9,000 molecules which have been selected. We collected the information about the molecules which have been tested toward the glucocorticoid receptor NR3C1. Okay, and those are the ones which are highlighted either in blue or in orange on this chemical space. Okay, so the gray or all the others which were not tested. So for at least we couldn't find any information in the public domain. So for those who are either in blue or in orange, they were tested, and the orange were found to be active toward this nuclear receptor. The blue were found to be inactive. Okay. So this is the chemical space when it comes to the this glucocorticoid receptor. So then you can guess what we did was we had a look at the response into the different cell line. And just to give you one example, I mean 2 examples basically, this is the result we did obtain into the A549 cell line, where most of the active molecules toward this nuclear receptor were found to be located in this cluster in this biological space, like it was the case for at the chemical level. Yeah, sorry, I forgot to mention that. You see that most of the molecule which were active was located in this cluster there on the top left of the chemical space. And the same was true in the biological space. So most of the actives were located into this this cluster in this cell line. When you look at the another cell line, the MCF7, the situation was completely different. Okay, you have the positive molecules toward this nuclear receptor was scattered all around the biological space without any kind of cluster and cluster visible into this representation. So from that, what we have concluded is that actually compound in that case with molecular, I mean structural, similar chemical structures can induce similar responses, but this will be depending on the cellular context we are talking about. And speaking about that, it's certainly due of, but we know that in the case of MCF7, but it's also about how much of the pathway of the genes involved in the pathway of this nuclear receptor is expressed into these cell lines, in the different cell lines. Okay, so we knew basically that in this MCF7 cell line, this nuclear receptor was not expressed at all. Another example, following the same type of visualization. So we did have a look at tubulin inhibitors. In that case, you see same representation, positive and negative. So the positive molecules inhibiting the polymerization of tubulins are spread all over the chemical space. But now, if you look at the gene signature and in the biological space, you see that there are most of the active molecules are clustered into a really specific area of the biological space. And by the way, these genes which are expressed in this cluster in the different cell lines share a lot of similarity, and they have common pathway which has been activated. So in that case, we can see that similar biological responses can be induced by compound with diverse chemical structure. So I wanted just to show you this type of examples to illustrate how much the cellular context and how much the test system in vitro, which has been is selected to perform the evaluation, is important when it comes to categorization of the molecule or to gain information about the properties of new chemical entities. I would like now to move to the second example. The second example is about is dealing with cell painting. So it's an essay which have been developed in the laboratory of Stuart Scherber and Anne Carpenter. It's an unbiased essay, and in that case, there are different dyes which are the capacity to stain different subcellular compartments. Could be the nucleus, the ER, mitochondria, actin, Golgi, plasma membrane, nuclei, and cytoplasmic RNA. And out of all those images, so it's a multiplex assay, you can retrieve several thousands of metrics of each image cells. And it has been shown that this assay can be used to study biological impact and of molecular and genetic perturbation of molecules. It can be used for the grouping treatments based on similarity. It can be used from compound target identification and mode or mechanism of action prediction. So in a nutshell, this technology has been shown to be a good and an excellent, I would say, tool to get a cheap biological rich information that can be used to support the chemical space exploration and to some extent also chemical optimization. So in this type of types of technology, there are very specific steps and workflows to make this analysis of this data. So the first thing is about feature extraction. So you start with the image, you make some quality control, so you extract the features. You have to do some dimensionality reduction, and then you can play with the features or the patterns of the different chemical entities in the test system and apply that to many different types of biological question. Could be on heat discovery or could be on tox, or hopefully it will be the case in the near future. This is one example that has been published by a colleague in the US EPA in the paper from Nifeller et al. In 2020, where you have the pattern induced by 4 different, it's too small, I cannot read the name of those molecules. Basically, you have there the different features from the nucleus, the cytoplasm, the endocrine reticulum, and so on and so forth. And the different you can see the different types of signatures that the molecule are inducing at the phenotypic level that are that provide the pattern actually of the biological effects induced by the molecules. So in terms of conclusion and key learnings that we could make out of these few examples is that gene expression and cell painting signatures extract helpful information from biological responses induced by chemicals. One can make hypotheses in terms of MIE when it comes to new chemical entities based on the evaluation of the similarity between this new molecule and the reference compound, and that could enable us to make this what we call biological space exploration. I didn't, for the sake of time, I didn't show you that, but we did apply this, we did use this type of data to make some predictive analysis based on machine learning, based on this signatures model. And we did show that they could be as performant as QSAR models to predict the potential of a new compound to interact with non-targets, and this without the limitation which is associated with the QSAR model when it comes to the chemical space limitation. Yes, so I will now move on to the next part of my talk, to briefly touch upon some work we have been doing about Denovo design of heat molecules based on biological space data. And just to start with, so this work has been done by my postdoc Oscar Mendes Lucio, who is now working for J&J. And I start to try to explain you, the big difference between the classical QSAR model and the generative model that has been used in this work. So in the classical QSAR model, you start with a set of molecules, a set of effects of this molecule, let's say in that case of a names model, names assay. So you have the so-called training set. So you have a series of molecules with an annotation about the positivity of the negativity of those molecules toward this AIMS test. Okay. Using this training set, we train a machine learning model, and once it's done, one can use a brand new molecule and ask the machine learning model to predict what's going to be the effect of the molecule if it is positive or negative into this assay. Of course, if it is positive, then you go back to the chemist and say, yeah, please do something different and provide me with a negative AIMS molecule. So this is for the classical QSAR. When it comes to the generative model, we have also in that case molecules and a training set. So, but the training set in that case is composed of in that case gene expression profiles. So you have a set, and in that case it was a very large set of data where we have the molecules and the in vitro gene expression profiles that were used to train a model. In that case, it was a deep learning model, and using this model, then the whole input was completely different, because we started from a desired gene expression profile that could be also a desired morphological pattern, and out of this desired gene expression profile, which has been used as an input, we asked the model to design a brand new molecules out of it. And yeah, in a nutshell, this is the overall principle. So it brings us to the concept of safety by design. So and that's essentially something we have in mind to develop. So we did this proof of concept of applying, in that case, thousands of chemical compounds in vitro cell culture. So in that case, it was cell lines. We did use the gene expression data or the cell painting data to capture the biological responses induced by the different compounds. And this model has been used. A model has been used to learn basically the relationship between the chemical structures and the gene expression or the cell painting profile to enable us to make this, as I said, input desired gene expression profile or high content analysis to design new, in that case, new drug hits as a proof of concept. But on the long run, of course, we want to design new compounds with an optimized safety profile. So I went rather quickly on this topic, but I wanted to mention it. So as a conclusion and key learning, so we provided the first evidence that it is possible to guide chemical design using either gene expression or cell painting data, this coupled with machine learning. So the de novo design of chemicals seems possible based on biological effects. And we are at this stage where we are making the experimental validation of a set of machine learning design compound at the moment. On the long run, we can then envisage to de-risk compound by chemical de novo design based on specific signatures related to toxic molecules and somehow wipe unfavorable safety profiles. And I will now move on to the last part of my talk. Something I would like to propose in terms of cross-sector collaboration. So when we look at the moment about at the different ways to perform the risk assessment, in particular for plant protection products, and you see I'm a very big fan of triangle, so another triangle there. So we start from in our pipelines and R&D development. So we start with in silico, in vitro studies, then we move on to subacute, subchronic, and then apical studies. And typically, in the actual paradigm, it's mainly the apical study which are driving actually the condition of registrability of our actives. And so for this, we apply in this current testing paradigm, what we call this top-down approach. So we start with the adverse effects characterization. We look for the animal MOA, then we derive some mechanistic information, which then allows us to make this human relevance characterization. And as we know, there are also at the moment, and since now a decade or so, this new paradigm which is emerging, which is a bottom-up evaluations. We start from in vitro and in silico studies, and based on high throughput in vitro testing, collection of MOA, AOP, toxicity pathway information, coupled with quantitative in vitro to in vivo extrapolation, then we aim at predicting the adverse outcome directly into the humans. Still a lot of things to be done and to be defined to make this really possible. The intention there is to say, let's have a look if we could already, as a first step, make this bridge between this high throughput in vitro evaluation with adverse effects which have been already observed in tox studies, in laboratory animal tox studies. And that's basically the rationale of this proposal. In this proposal is based on the following facts, that we are now at the stage where the technology is now available to make and to perform large-scale biological exploration that can be used for MIE prediction, read across, point of departure determination, and possibly in the future for de novo design. So let's see if it will be worth to explore the value of the collaborative efforts associated in industry and academia in order to build a shared data set of gene expression and or morphological pattern derived from in vitro based test systems. As a first idea of implementation, the idea would be to start with the characterization of the transcriptomic and the morphological pattern of a large set of compounds for which we can anchor the results into an in vivo tox profile. So, on the first place, we were thinking about the rat 4-week studies because it could make some sense, and there could be some joint interest with the colleagues in pharma, I would say. And in a nutshell, for this, we will need to agree on the test system. Could be the cell line, the primary cell culture, I mean the test system itself. To agree to provide a few milligrams of test compounds with their tox profiles, including ideally the IDME profile of the molecule, and run the experiment in a central facility to ensure the comparability of the results. And also to agree to share the results among the consortium members. So this is something I tried to push forward as an idea, and I would be very happy to get your feedback and to see if it generates some sort of interest to you who are attending this call. And with this, I would like just to finish and thank you all for your attention. And I'm really happy to answer your question or to comment about this proposal of collaboration. Thanks to you all.
Thank you, David. This was a very, very interesting talk and technology. I'm going to open it up to see if anyone on the audience have any initial questions regarding any of the parts of the talk. Feel free to also post any questions on the chat box as well. If anyone has a question, please unmute yourself and go ahead and ask them.
Hi, David. Richard Curry here.
Hey, Richard.
Just a question. When you were doing your generative models, you kind of glossed over how you defined what a desired gene expression profile might look like. So how did you design and describe what a desired profile was?
Yeah, in this proof of concept, thanks, Richard, for this question. Yeah, I went through it quite quickly, but in this proof of concept, what we have done is that we have the luxury to play with a series of knockout cell lines. Okay, so we had the gene expression profile of many different knockout cell lines, and in particular, knockout cell lines from pharmacological targets. And so what we did was to make the hypothesis that the transcriptomic profile of the knockout of a pharmacological target could be somehow similar to the profile of a to the profile of a potent and specific inhibitor of this target. Okay. And it seems to work. I must say, it seems. I mean, we are very first evidences, but it seems that it works. But on the long run, when it comes to the tox and the safety aspects, that could be the idea would be certainly different. We would have to identify and characterize the patterns which are known to be associated with tox outcomes, and ask the model to kind of I use the term wipe, but it's probably not the best scientific word with verb for this, but to optimize the chemistry against that, actually.
Yeah, I think it's an interesting thing because most of these generative models work as you just described it, trying to get towards something, right? You know, there's a goal in mind, and removing toxicity is probably moving away from a set of profiles. And I've not really seen a proof of concept to prove that a model can actually move you to a place that's not where you started from, if you see what I mean. So Just don't give me all of these things. It's a very different thing to give me something that's a bit like this.
Yeah, and of course in our case, I would say in agrochemical, but I think in pharma it would be the same. Is that of course there are another big challenge is about being able to. I mean, it's multi-dimensional, this thing. Huh? So one has to optimize the molecule for the safety profile, but at the same time not impair the properties when it comes to the biological efficacy and on the targets itself. So on the insects, on the plant, on the wheat, and the very same probably will be true for the colleagues in pharma, where you still need to get the pharmacological effects. And in that case, it could, I mean, in terms of applications, it would be mostly for the off-target effects, probably where you could think of using it. But again, it's still at its very first infancy. I would say we still have to do plenty of things in order to make sure that this thing will work. Yeah.
Yeah, you probably need your collaboration data sets to before you can begin to build those kind of models anyway.
And that's kind of the intention behind this cross-sector collaboration, because on the long run, this thing could help to make this kind of repertoire of all the both transcriptomic and morphological patterns, which are if not causally associated, but at least associated with an adverse outcome.
Yeah, thanks, David.
Thank you, Richard. Chris Corton, you're next.
Hey, David.
Hey, Chris.
I'm assuming you can hear me, David. So I was really intrigued with your TSNE networks.
Yeah.
And as you may know, that HESI has a number of projects to build and characterize and use in characterization of AOPs, gene expression biomarkers.
Mm-hmm.
And there's a lot of parallel efforts going on in a number of labs. So I can see some value in using these networks to test the ability of these biomarkers to identify chemicals that are MIEs or key events in AOPs. Mm-hmm. I can also see their utility because you've looked across a number of cell lines. Mm-hmm. I can see their utility in determining the domain of applicability. Yeah. I'm wondering how hard it would be or how hard it is just to take well-characterized gene sets and then compare them within the TSNE networks to answer questions about what are the molecular targets of a particular transcription factor, and then in which cell lines do you actually see that?
Yeah. I think it's all about the what I would call the physiological relevance of the test system that needs to be on the first place very well characterized, and to make sure that actually the pathway you want to investigate is expressed in a kind of physiological manner that will make possible this retrieving meaningful information out of it, because otherwise it's just a risk to make the to shoot at the wrong target. It's a kind of metaphor there, but I think it's essential to look at the physiological relevance of the molecular effects which are derived from in vitro test systems, in order to make. In our case, we are really working at the very beginning of our pipeline and R&D pipeline in order to make sure that the decisions we are making in terms of compound to be promoted are really translating into the profile. As soon as you get into the animal, then you see what you expect to see, and not you're not surprised by something that your test system in vitro kind of was not able to see or to tell. Yeah.
Okay,
So,
thanks.
Thank you.
Thank you, Chris. Any other questions? We might have time for maybe one more.
I have a
Comment?
question. It is Alexei.
Okay.
Alexei Poteleshnikov. I have a question.
Yeah.
I wonder if you tried this L1000 system internally at your company, and what are your impressions about reproducibility of this network, especially if you go beyond the cell lines which are publicly available, for example, or especially if you add liver to the mix, because this seems to be like an obvious gap in a public data set.
You
I
mean
wonder
the
if you can.
metabolization? Yeah.
Yes,
Okay.
right. And well, just a general impression about L1000 system as a sort of transcriptional, and also how do you expect it to perform beyond the cell lines?
So technologically, I think the L1000 it was developed initially at the Broad Institute back some 10 or 15 years ago. There were at some point a startup company which was developed out of it, but it was stopped. We did explore the possibility to run some experiment using this technology, but finally we didn't follow up the too much, because it was not so much available, let's say. And so we now in the lab are relying on another technology called GoScreen from the so it's an high throughput DNA array-based technology, which now allows to run really a decent number of molecules at once, and is not at the same cost as what was claimed by the L1000 people initially, but still it's now realistic to think about applying that to hundreds or if not thousands of molecules in a screening mode. Yeah. So but of course there are also these RNA-seq methods which are already also there to do this type of things in an effective manner. I mean, it's now you even don't have to make the RNA extraction of your samples. You expose your cells, you wash them, and then you lyse them, and then you run for the gene expression profiling right away without any RNA extraction steps, or so it's really convenient.
Right. Yes, thank you. I mean, that's our experience was similar. At the end of the day, we didn't pursue L1000 beyond, some initial experiments.
Yeah, I'm not really clear what will be the future, or if there will be some. I mean, I don't know if there are some activities with the L1000 at the moment at the board. Frankly speaking, I'm not aware of it.
Mm-hmm.
Thank you, Alexei. All right, we're 2 minutes away from the top of the hour, so I just want to use this time to thank David for a great presentation and for your proposal at the end. And I also thank you for that proposal for collaboration. I think, as Chris Horton pointed out, this draws quite some parallels with some of the research that's ongoing at HESI. So we will definitely be following up with you with that regard. There's quite a bit of interest, and I'm really look forward to potentially having this being proposed, and we will be following up. So I also want to invite anyone else who didn't get a chance to submit a question or a comment to contact David directly or myself through email, and we'll make sure to get those answered as well. Thank you so much, everyone.
Thanks to you, everyone. That was a pleasure for me. Thanks a lot.
Thank you,
you.
David.
Thanks, everyone. Thanks, David.
Yeah, bye-bye.
Bye.